previous session, I talked about that how simple phenomenon of organogenesis and somatic embryogenesis have been exploited for benefit of improving the crop quality and quantity. For example, we talked about micropropagation, how it facilitated the asexually reproducing plants to be multiplied and creating homogeneous genetic stock of the plants which were better performers in the field, which is normally done through cutting processes and other means. Uh, the advantage of micropropagation, as I discussed with you, is that you can raise these kind of plants all through the year if there is no limitation of the favorable climatic condition to be there. And also, you can raise these plants through the source, which is disease-free and also better performer. So you can generate the plants which are better performing in the field. And also we saw that how somatic embryos after encapsulation can be a source of conserving the better plant types, and so now see, when all this was happening, the efforts were made uh, whether the organs also can be cultured. So far what we saw, the explants source tissues were taken as either from stem tissues, leaf tissues, and then they were made to de-differentiate and redifferentiate either by organogenesis or by somatic embryogenesis, you know. Then in around 60s of last century, the efforts were made to uh, see if the organs can be cultured and what happens when they go for organ culture. And there were two objectives. One is to study the developmental process in vitro. Okay, so uh, the floral parts of the plants was taken, and in this case is the anthers or the ovules, you know. So when these efforts were being made, the success was, I mean, mostly the plants regenerated were from the somatic tissues of the anther or the ovules. In the anther, the pollens are located uh, inside the anther, and pollens are the haploid cells or the reproductive cells which participate in sexual reproduction. So when anther culture was tried, most of the plants originated were from this diploid tissue which surrounds the pollens or pollen sacs, and they were all diploid in nature. But accidentally, what was observed that some plants were different than the other plants. And how they were different? They were very weak. And when the chromosome number was checked in these plants, they were having half the chromosome number, and that then gave rise to another concept that by taking the reproductive cells, one can regenerate a plant which will have half the component of chromosomes. For example, if an organism has 20 chromosomes, then these plants have only half, that is 10 chromosome number. So we will dwell upon this event which happened, and it was a breakthrough in the field of plant crop improvement. You know, so um, we will go through uh, very systematically that how this discovery was made, and and what was the implication of this discovery. Okay, so I'm referring to this dihaploid production or formation of production of dihaploids, or first the haploid and then the production of dihaploids. Okay, so let us look at what is the haploid plant. Okay, so as a definition, a haploid plant has a single set of chromosome. You know, means they have n number of chromosome in its cells. Okay, so if the diploid cell has 2n chromosomes, these plants, the haploid plants, have only half the set or single set of chromosomes. You know, so a haploid plant has single set of chromosomes. You know, in a diploid plants, you have two sets of chromosomes, and in haploid plant, you have half the number of chromosomes. Okay, so if the diploid cell has 2n number, the haploid cell or haploid cell will have n number of chromosomes. So if the 2n is 20 in a diploid, in haploid, n would be 10. So that's how haploid plants are defined. And as I mentioned, uh, this discovery took place in 60s of last century, and the lab doing it was the one from India, like Guha and Maheshwari. These are the people who first um, demonstrated that it is possible to culture anthers and raise haploid plants, you know, in Datura species. The experiments was done with Datura species, and they could recover plants which were having half the set of chromosomes, or n number of chromosomes, as compared to 2n number of chromosomes. And this technique was subsequently refined by niche and niche in tobacco systems. You know, so these were all experimental systems which were tried uh, initially, like in the Datura and then in tobacco, and then it was suggested that it is, if it is happening in these two model systems, it can happen anywhere. You know, and the concept is that uh, if the anthers are cultured uh, uh, properly, given the right kind of environment, physical and biochemical, and media and the growth regulators, it is possible to raise haploids through pollen culture. Okay. Now, how the haploids are produced, as I mentioned, one is by anther culture or microspore culture. You know, you can directly take pollens, or you can directly take ovule from female reproductive part and culture it to get haploid plants which have n number of chromosome, or half the set of chromosomes as compared to the diploid counterpart cells. Another method which was tried was trying an interspecific interspecific sexual reproduction. For example, Hordeum vulgaris is one species and Hordeum bulbosum is another one. See, the sexual reproduction doesn't go in the same species. Okay, so that's how the speciation takes place. That sexual reproduction is limited to the same species, and it is not possible to cross two different species sexually to get that. Now, here, what happens when you cross those and get an embryo? So, in this, what happens? the growing embryo tend to eliminate one set of chromosomes, coming from one species. And this was then helped, this elimination of one set of chromosomes by the in vitro cultures, a technique what we call embryo rescue. Otherwise this embryo will die. It won't grow into seeds. But if you take out this embryo and rescue it in vitro by culture, then it is possible to rescue it and generate the haploid plants. So this was another alternative method of producing haploids experimentally first. Now, what happens here if you see diagrammatically? See, this is Hodem vulgar, which has 2n, 14. The chromosome numbers are 14 here, and Hodem bulbosum also has the 14 chromosome number. So when they are crossed, you know, and the embryo is rescued, you have haploid barley plant, which has this 7 number of chromosomes coming from the Hodem vulgare, and Hodem bulbosum thing is eliminated, when you do embryo rescue, you know. So this was once a very routine method of producing haploids, but um, in the improved methods of culture, pollen culture, and ovule culture or anther culture, it is now routinely done by anther cultures or the microspore cultures rather than going by this interspecific crossings and doing embryo rescue. So popular approach subsequently, when the techniques for anther culture improved and routinely it was done, then this approach was followed further, in future stages. So we will talk about this. Now, what happens pictorially, what I had been talking about for anther culture or the microspore culture, you take out the anther from a plant like this and put it on the culture media. Okay. Now, this can have two routes of differentiation. One is by callus formation, and then the callus gets differentiated into haploid plant, plantlets. Or it can generate embryos, and these embryos can directly give rise to a plantlet, shooting and rooting the express. So that's how, this is achieved. So this is just a pictorial representation of the sequence of events taking place here. Now, if you see what are the factors which affect the anther culture, this is by and large the same factors which we saw, which affects the normal tissue culture events. For example, genotype. Now, genotype is the one which affects the in vitro techniques. Okay, what I'm referring to, genotype of the host plant. As I mentioned, that some plants are more amenable to tissue culture manipulations as compared to the others. Okay, so and then the growth of the mother plant. So mother plant has to be healthy, growing in right kind of conditions, so that when you take the part for in vitro manipulations, the parts are also healthy and they respond to tissue culture techniques slightly more positively than others. Then the correct stage of development, you are picking up the anther. That is a very, very important factor for anther culture, that you should pick up the anther at the right stage of the development. And these things need to be optimized. You know how to find out what is the right stage. So when you see the anther formation it has taken place, and then inside the anther you have the pollens growing, the pollens have been produced, and then you pick up one. Okay, so all these kind of things have need to be optimized, and to pick up the right kind of to pick up anthers at the right stage of the development, so that when it is taken to in vitro, it responds positively. And then The pre-treatment, like seed germination, you have to pre-treat them with cold and heat and other sorts of treatments for effective germination. Similarly, here, the pre-treatments may be required, may not be required. So one has to optimize this also. Sometimes cold treatment helps, sometimes heat treatments help, and the culture media. The culture media is also a very, very important factor other than the nutrients, uh, the semi-solid nature or the floating kind of a things, what kind of, how much agar you are using in to solidify that. That is one factor. The growth regulators, the kind of growth regulators one is using, it needs to be optimized, you know. So these are the factors which need to be looked at, both from the explant side and the in vitro technique side. You know, all these needs to be optimized. Similarly, the ovule culture, you can produce haploids using using ovules or the female reproductive part, and basically it has the same factors which I have discussed for Anther culture, and these factors are pretty common, you know. The question is, if you have an haploid plant, what do you do with this haploid plant? You know, so normally, since they have half the number of chromosomes or one n set of chromosomes, they are very weak. And also, they are sterile. If these plants grow up to maturity, they cannot sexually reproduce. Why? Because they are haploid and they cannot produce the sex cells. Because to produce sex cells, the plant has to be diploid. The microspore mother cell is a diploid cell which undergoes meiosis, and mitosis to give you the four microspore or the pollens you know so so therefore these haploid plants, when generated, they are sterile. They are not going to participate in reproduction. Okay, so normally what happens when these plants grow, they spontaneously undergo the doubling of chromosomes, or what we call the diploidization, or they are called as dihaploids, because this half set of chromosome is going to be duplicated as it is. Okay, so haploid, dihaploid. So you have now two set of haploid chromosomes. You know, so they are referred as dihaploids. When a haploid set of chromosome gets diploidized, we call it as dihaploid formation. You know, so dihaploid formation is important so that this plant can participate in future sexual reproduction. So how that happens, this diploidization, how the single set of chromosome gets converted into two sets of chromosome. So that happens spontaneously, and also it can be induced by chemical called as colchicine. When you spray colchicine to the plant, the diploidization takes place. Okay. Okay, so once the diploidization has taken place and the plant is now a dihaploid, then it is like any other plant, which has the two component of chromosome, diploid, or two sets of chromosome, diploid. Okay, but then what is the benefit, and how this process has really helped in crop improvement? That is the question. So as a technique, it was developed. People could be able to culture anther or ovule and generated haploid plants which had half the chromosome number, or n set, or n number of chromosome as against 2n number of a diploid cell. And these plants could be diploidized to produce dihaploids, and they were fertile plants. As I mentioned, haploid plants are sterile. They cannot participate in sexual reproduction. So what is the application? How this discovery actually helped in crop improvement programs? Let us look at this. So now what happens when you have half set of chromosome or and then when it gets diploidized? So one thing which happens at the genetic level is homozygosity. You know, uh, all the sets of genes which are present on half set of chromosome when diploidizes, you know, then you see the all the genes become homozygous. If it is dominant capital T, it becomes capital T in the Mendel's work, if you refer to. And if it is a recessive gene, small t, then it becomes small t, small t. So in one go, heterozygosity is removed completely, and all the traits which are represented in the genes, they become homozygous. Now, what is the significance of getting homozygosity is that, as per Mendel's genetics, if you see the characters which present or the traits which are present in an organism is in three forms. Either they are homozygous for the trait, that is capital T, or they are capital T, small t, or small t, small t. Okay, so in an organism, you have homozygosity as well as heterozygosity coexisting, and that is the basis of variation, genetic variation in subsequent generation. This capital T, small t always tend to segregate in subsequent generation, whereas any trait which has come into capital T, it is fixed, and it will be taken in the next generation as capital T, and small t. So genetic variability is seen because of heterozygosity, and that is the point of concern. But what is happening here, connecting it to this new discovery, that the anther culture, when it diploidizes or made to diploidize, you achieve homozygosity for all traits, and that is the biggest advantage. Otherwise, this homozygosity takes longer time if you keep on breeding, if you keep crossing and by inbreeding, it takes a lot of time. So this was one of the very, very important discovery or use of this discovery of achieving homozygosity in one stroke, which otherwise takes. See, if you see here, normally, if these are the two parents, you have F1 generation generated, and then it has to undergo at least 6 to 7 cycles before you achieve the fixation of a trait. That is, the trait is becoming homozygous so that it won't segregate further, So it takes 6 to 7 generations. It means 6 to 7 years one has to work. But then if you come to the anther culture mode, here you just take the anthers from here, and then you have the thallus, green plant threads here, and then you have the diploidization in one go. So we are reducing the time in terms of fixing a trait in a population for a plant, it's 6, 7 years against 1 year. And and it takes another 6 to 7 years to release the variety. From this stage, you take another 6 to 7 years to release the variety. So total time taking to release the variety is 12 to 13 years. It's not simple. But then once this discovery was made, it could be possible to reduce the time of varietal release by 6 to 7 years. If you see here, in one go you get the trait fixation, homozygosity is achieved, and then another advantage which one achieved here is the homozygosity for recessive characters, which is still tougher. Dominant characters you can select and inbreed and select and inbreed and so on. Recessive characters are very difficult to work with, you know. But here, with this technique, even recessive mutation or the mutants could also be could also be made diploid and could be made or could be fixed in the sense that once they are fixed, they express. Means once they get diploidized, homozygosity is achieved, then the recessive mutation will also express itself. You know, so that is the advantage of getting homozygosity for a dominant trait as well as for a recessive trait by anther culture and then subsequently diploidizing it. You know, so that is the if we look at what are the achievements. So utilizing this approach, starting from late 60s until date, a lot of varieties have been released in economically important crop plants and model systems where it was first tested. Rice, wheat, barley, you know, and a lot of varieties have been released, and predominantly by China for rice and wheat. China took a lead in this and developed technologies for anther culture, hybridization, and then using these parental lines for their inbreeding purposes for their own known cultivars. So, and fixing the traits which they wanted to improve. And then, of course, it is being uh, done globally, actually. This approach of crop improvement or varietal releases using the agriculture hybrid. So, as I mentioned, the applications is introducing homozygous dihaploid lines, and they may be, I mean, they have been useful in isolating mutants, dominant mutation or recessive mutation. When it comes into homozygosity, it expresses itself. You can choose it and then use it use it for the crop improvement programs. You can do genome mapping by this, you know, and then you can look for different traits like the disease resistance or the performance against abiotic stress factors, abiotic factors, which are seen to be affecting the crop productivity in the field and also the nutritional values. So the field is wide open for its application. You have to just pick up the right kind of mutation and then take it for further breeding purposes. Limitation is the technique as a whole, in the sense that the anther culture in vitro manipulation should work in the lab by the researcher. Now, that is where the whole thing, as I mentioned, there are so many factors which affect the successful culture of anther, and this needs to be optimized. And unfortunately, tissue culture protocols, whatever has been defined by in papers or in literature, whenever somebody starts working in the lab on a new technique or on any technique of in vitro plant regeneration, they undergo a rigorous optimization protocols. So this is the main limitation, that you just cannot pick any, enter from any plant and follow some protocol and you end up with a haploid plant. It doesn't work like this as far as tissue culture is concerned. Not only Anther, but anything which so far I have discussed and subsequently what I'm going to talk, this needs to be optimized. So this is one limitation, and in some plants it has not been feasible to produce haploids, or some whatever optimizations were done, but it could not be possible. So, what I have tried to do in this session is to give you an idea about yet another application of plant tissue culture technique, that how it was exploited for crop improvement programs. Okay, so with this, I stop here and continue in next session on another interesting aspect of plant tissue culture. Thank you very much for